All right, so this is our overview for this topic, right? Last time we talked about anatomical barriers and things associated with them. We talked about different receptors, and we're going to continue on that and go over the complement system today. We talked about cytokines and phagocytic cells as well as other white blood cells. We've previously talked about inflammation, and hopefully we will finish up with fever today. So, in a little bit of review, in addition to these barriers of your skin and your mucous membranes, and of course I erased the whole thing, but that's okay. Lysozyme, right, which is an antimicrobial substance found in your saliva, found in your tears. What does this destroy? What does lysozyme destroy? Peptidoglycan. And where is peptidoglycan found? The cell wall of bacteria, and a lot of it in gram-positive bacteria, right? Lactoferrin. What is its job?
Binds the iron.
Binds to iron, right? And it keeps it away from the bacteria, again, because remember, they need it for their metabolism as well. Defensins, what are those?
Form pores in the microbial membrane.
Right, form pores in the microbial membranes, procols. So Carol, you're done. You've answered at least 3. Okay. So then we get to our cells, right? And we have groupings of them. We have granulocytes. Who are the granulocytes? Their names all end in "phil." Right. Neutrophils, basophils, eosinophils. Your mononuclear phagocytes, there's 3 of them, but one of them really is just a differentiation, a precursor to the other 2. So what is the one found in your blood? Monocytes. What's the ones, what do you call it when it differentiates in your tissues and stays resident in those organs? Macrophage. And what are the ones that gather antigens and are going to bring them to your adaptive immune? Dendritic cells, right. What are our 3 lymphocytes? And natural killer cells, right? B cells, T cells, and natural killer cells, right? And then dendritic cells, as I said, because they're so cool, they kind of sometimes are even considered in their own category. So now let's talk about their functionality. So I've already said dendritic cells are going to gather up antigen and present, right? What do macrophages do again? They eat, right? And this is happening in your tissues. Mast cells are important in eliciting inflammation, again, in your tissues. This has to do with the granules found in their cells. Which one of these guys is a close relative because of the similarities of their granules? Eosinophil, basophil, or neutrophil to your mast cell? Basophils, right? All of these guys will be found in your blood, although you can't differentiate between the lymphocytes in your blood. They all pretty much look the same if you do like a differential test. These are the main components of your adaptive immune, especially your B and T cells, so we're really going to get into their functionality later. What's an important function of your neutrophils? What do they do? They eat bacteria. They're phagocytic cells, right? And they're only enter into your tissues during inflammation, right? They're going to be called there. Where these guys live there, right? Your macrophages, your dendritic cells. Basophils. Allergic reactions, inflammation. Eosinophils. Parasitic worm infections, also sometimes with allergy and inflammation. And again, it has to do what's in those granules, right? And we'll talk a little bit in detail about basophils, but really not going to get into eosinophils, right? So functionality, right, and classification. So surface receptors are how we able to know when stuff gets past those barriers, right? Gets past the mucus, gets past our skin, gets into our tissues. The cells themselves have receptors on them. And then the cells communicate with each other via the cytokines. And there's several different classes of cytokines that you need to know, right, what they generally do. We're not memorizing individual cytokines. So the surface receptors on the cells are almost like their eyes and ears, and they actually even have internal ones as well. The cytokines are those chemical messages, they're the voices. And then the adhesion molecules, right, help them adhere. And we saw this in inflammation, right? How those white blood cells were able to adhere to the endothelial walls and then attach and be able to move into the tissue where they're being called. So we talked about these guys last time. I'm not going to review too much of that right now. And I think we ended on adhesion molecules last time, right? So now we're getting to the new stuff. So what happens, an outcome is what happens, right? When toll-like receptors are engaged, when the complement system is activated, when there's detection of long double-stranded RNA, right? And double-stranded RNA, is that us? Do we have double-stranded RNA? No, RNA is single-stranded, isn't it? So where would double-stranded RNA maybe come from? Viruses. Nasty little buggers, yep. So we have systems within our blood that detect signals of tissue damage or microbial invasion, right? We have to be able to sense that they've gotten in. And we rely on patterns, right? So these similar molecular components of the invaders, right? Over time, evolutionary-wise, right, we've developed these receptors that recognize general patterns in the microbes, right? Whether it be viruses, bacteria, protozoa. And so that's going to help us trigger the alarm, right? So they're sensing it. When it's detected, right, we're going to call up our host defenses. So they have lots of horrible acronyms, right, that you'll sometimes see, right? By no means do I want you guys memorizing them, but the important thing here is that it's receptors, right, and that we're looking at patterns. So whether they're molecular associated patterns or danger associated patterns or pathogen associated patterns, there's a pattern here, right? They're all patterns. So let's look more specifically, what are the types of patterns that are common, right? So cell wall components of bacteria, lipopolysaccharide is the outermost covering of what type of bacteria, gram-positive or gram-negative? Negative, because it's part of that outer leaflet of the outer membrane, right? Peptidoglycan would be the outermost covering of gram-positive. Lipotectonic acid is associated with gram-positive. Positive for that one. Lipoproteins could be any bacteria associated with their cell wall. So these are general components of general groupings of bacteria, right? General group of gram-negative and gram-positives, where there are specific ones out there, right, that have specific abilities, like E. coli 0157, right? That's a very specific strain of E. coli, very pathogenic strain at that, okay? Our innate immune system would just recognize it's a gram-negative bacteria, right, because of its lipopolysaccharide. It wouldn't recognize its specific sugar components that are associated with that pathogenic strain. That level of detail would be done by our adaptive immune system, okay? So here, just these general patterns. So other things are like flagellin, which is the protein that makes up the flagella of bacteria. Viral RNAs, right? They're distinctly different from our own RNAs in that they can even be double-stranded where ours are not, right? So we have receptors, so as I said, not only outside of our cells, but also inside our cells for these general patterns, right, that are, of course, a danger signal. They're microbes, right? They could even be pathogenic, disease-causing. Still with me? Okay. So toll-like receptors are a group of these pattern recognition receptors. And we have them not only on the surface of our cells, but you also have them inside of our cells. So notice the receptors, they were just proteins embedded in a membrane, right? And so what do they recognize? They recognize certain molecular patterns, right? So they've found that toll-like receptor number 1 recognizes lipoproteins, right? And notice that toll-like receptor 3 is recognizing viral double-stranded RNA. And where is that receptor? Inside the cell, because that's where that component of the virus is going to be accessible when it gets inside of our cells, right? That the DNA, because remember, the RNA, they have a protein coat, right, when they're outside our cells, but when they go inside of our cells, they'll lose that coat, or the nucleic acid will come out of that coat so that it can be copied, right, so it can take over our cells. So that's where we need that detection. And notice it's attached to a vesicle, it's a membrane, commonly referred to as an endosome.
So you said we didn't have to memorize what
You don't need to know, like, lipoproteins is toll-like receptor number 1. You need to know these general examples of what type of patterns we're recognizing, right? So we're recognizing, lipopolysaccharides. And you guys already know, right, as you repeated back to me today, that would help us against gram-negative infection because that's a component that they have. Okay? Right. And so in this animation, one of the reasons why I'm not playing it is that they talk about this process, right? And they go into real detail about, the different molecules involved in carrying the signal. But these are receptors, and that's what they do, is they interact with specific molecules, right? They only bind to specific molecules. And they're going to carry the signal inward. So notice it's interacting, right, with the receptors. Other molecules inside interact with the receptor, and they're going to carry this signal usually all the way into the nucleus and interact with the DNA. Because remember, this is the command center. This is where the information is housed. So depending on that signal, this cell is going to respond in some way, right? Like if I was to yell fire right now, everyone would leave the room. That's the common response to that signal, right, to that stimulus. So the same thing here, right? These cells are going to respond based on that signal, right? They're going to do something. Most commonly, right? So we're going to have expression of genetic information. The most common response is an inflammatory response, right? Obviously something bad got in, we've got to call all the rest of the white blood cells to the area to help us out. How is that cell going to elicit the inflammatory response? It's going to release cytokines, right, chemical messages for our body to be able to sense what's going on, and those white blood cells come to the area. So definitely, of course, our sentinel cells, the macrophages and the dendritic cells, have these receptors on their membranes. Other cells do too. And as we said, even inside the cell, they'll have receptors for stuff that gets inside. So notice even bacterial DNA for intercellular bacteria, right? Single-stranded RNAs that aren't ours, right? Double-stranded RNAs, which definitely aren't ours. And so depending on the signal, so, definitely if it's bacterial, we're going to have an inflammatory response. If it's viral, there we have particular mechanisms for viral response, right? So depending on which receptor is triggered, there's going to be a different response. But one of our most common responses and outcomes with infection, right, with invasion, is inflammation. Getting the white blood cells to where they need to be to help us with the invasion.
Do we need to know, like, which of these pattern recognition receptors are, like, inside the endosome and
Right.
on the cell membrane instead?
Pretty much just the nucleic acid ones are inside the cells. So as a generalization, but I wouldn't ask you guys to that level of detail, like where is the double-stranded RNA receptor? Is it inside or outside the cell?
Said that for 200 level.
Right, but no, I mean, like, you guys should be able to make an educated guess anyways, right? Nucleic acids are inside cells, right? We wouldn't have receptors for that on the surface of the cell. Okay, moving on. All right. So NOD-like receptors are found in the cytoplasm, okay? So these aren't necessarily attached to endosomes. And again, this is for when stuff gets in. Some bacteria actually get inside of our cells, whole, intact, right? So we would need peptidoglycan detection even inside of our cells, not just outside the cells. Again, what's the most common outcome? Inflammation.
What does NOD stand for?
I don't remember,
Fair enough.
right? So NOD stands for something. And this is why I hate immunology sometimes, is we use these acronyms, right? And we get to the point where we start to forget what they stand for.
The military is even worse.
Oh, yeah. Trust me. I know. My first husband was in the military, and when he came back from basic training, every other word was an acronym, and I was like, stop.
The
Okay.
Navy does that. I swear.
What's this one, this one, this one, this one? It's like learning a whole nother language. And immunology is kind of like that sometimes too. And it's one of the things that I'm not so fond about, although I love this, this area of study because it's really fascinating, right? What our bodies can do. And we're just scratching in this course the level of understanding that we have. But in research in general, we're also kind of just scratching the surface. There's still so much we don't know about what's going on. It's a pretty amazing system, to say the least. So speaking of systems, the complement system is a system of proteins found in your blood and your tissue fluids. And these can help with detecting invasion, right? Because these proteins can interact directly with the bacteria, right? And can help elicit inflammation. They can even directly destroy the bacteria and even foreign cells that we'll talk about later. So notice we have a splinter, right? Some bacteria have breached the skin barrier because of that, have been pushed in. So some of the proteins involved in this become activated. And C3A refers to, it's a complement protein. It is number 3 in the order in which they were discovered, not necessarily the order in which they're activated. And A meaning it's a piece of that protein. So that protein actually breaks into 2 pieces. This particular piece elicits inflammation. This is a cytokine in itself, right? This protein in this system. This is one of the few you actually need to memorize its letters and number, right? C3A. And I will say it over and over again throughout this course, kind of like gram-positive and gram-negative. The next one is also involved in inflammation. It's C5A, but this one specifically attracts the neutrophils to the area. And remember, they only travel into our tissues during inflammation. C3B is the other half of C3A, right? It broke into the other half of C3, right? It broke into A and B pieces. B, I always remember B for binding in this one case, right? C3B for binding. As we'll see in a moment, it literally binds to the surface of the bacterial cells. When it does that, it's like tagging that cell for destruction. We refer to that process as opsonization, right? There's one other molecule that our body produces that's an opsonin, and that are antibodies, right? Whenever an antibody binds to a cell, whether it be a bacterial cell or fungal cell or another cell, it's tagging it for destruction. Phagocytic cells have receptors for these. Remember when we looked at phagocytosis? We mentioned C3b. It was on the surface of the bacteria, and the phagocytic cell had receptors, and they grabbed onto it, and they engulfed the bacteria. Now, the next one is a set of these proteins. This list I don't want you guys memorizing. It's not going to do you any good. But the name of the complex is important. So how cell lysis happens is via the MAC complex, and it's going to be on a later slide.
How do you spell that?
MAC. And I'm not talking about a Big Mac from McDonald's. I'm talking about a membrane attack complex. As you can see, it's a complex. It's lots of these complement proteins that come together. And you'll see they form a donut-shaped hole in the bacteria's membrane, right? Membrane. That's the important thing here in that name. And that causes the cells to lyse. So when complement becomes activated, we're going to get inflammation, right? The influx of white blood cells, even specifically neutrophils. We can get opsonization, tagging for destruction by phagocytic cells, the bacteria, when C3b binds. And we could even get lysis by the formation of a MAC complex. So, tissue damage has happened, right? C3a is going to bind to receptors on the endothelial cells, right? And that's going to help them become permeable. We're going to have those selectins and stuff expressed so that the phagocytic cells like the neutrophils and the macrophages can grab on, right? Head into the area. And they're going to go right to that area because they're heading towards C5a, right? Especially a neutrophil is very attracted to C5a. So it's heading over into the area. Notice the C3b is attached to the bacteria, and it's going to allow the receptors on the neutrophils to easily grab on for these tagged for destruction bacteria and engulf them and destroy them by phagocytosis. The other thing that can happen, oops, I fast forwarded too quickly. Here, let me just turn off the audio and play it. Is, as you can see, several of the proteins insert into this membrane and they arrange in a way that they form a donut shape hole, right? So there's a hole in it. So this one is forming. And as I said, it's commonly abbreviated MAC, M-A-C, but it stands for, and you need to know what it stands for, membrane attack complex. Right, and then the cells lyse. Now C3b doesn't always bind, right? The MAC complex doesn't always have a membrane to insert into and destroy. Now when we talk about bacteria though, who's got the outer membrane that this could insert into and potentially destroy them, lyse these cells? Gram-negative, right? MAC complex is very gram-negatives are more susceptible to that. Gram-positives, it would have to travel through the cell wall to get to the membrane, right? And it can do that sometimes, right? But it's definitely much more effective against the gram-negatives because their very outermost cover is a membrane. So what are the outcomes that could happen? We could have inflammation, remember, right? This is C3a and C5a are involved in the inflammatory response. Opsonization is by C3b. And lysis of the foreign cells, right, is by the MAC complex, membrane attack complex. So as I said, the complement proteins are numbered in the order they were discovered. Kind of goes in the order they're activated, not 100%. All the time in our bodies, C3 is splitting into those two pieces, A and B. But it doesn't continue on the chain reaction, right? This is almost like a domino effect or a cascade, they refer to it as, because once it's become activated, then it follows through. There's a certain step in this process when they're being separated that it will continue down that path. But that's the alternate, what we call the alternative pathway, which is sometimes kind of confusing. So there's those important ones you need to know, right? And this is even showing that phagocytic cell grabbing onto it. That C3b, that's a really good opsonizer. And there's our MAC complex. As I said, Most effective against gram-negatives, not so effective against the gram-positives, because it's tough to get to that membrane. There's a picture of it, right? All those black holes, right, are MAC complexes. That poor cell is toast. So when we talk about activation, right, as I said, there's a series of proteins. They're found in our blood and they're going to lead to when we become activating these things we just talked about. But how it becomes activated, there's 3 different ways in which it can become activated. We're talking a lot. And these are just the major proteins. There's other ones that are involved. Okay, so the classical and alternative pathways they talk about in this animation. This pathway is called the classical because it's what we discovered first. It is probably not what happens first, right? Because the activation is due to antibodies binding to the surface of an antigen, or binding to an antigen. An example could be a bacteria, could be a virus, right? Could be protozoa, fungi, foreign human cell, be any number of things. But antibodies are a product of our immune system. Unless someone has given them to you, right? You had to produce that. In order to produce that, the very first time you see a pathogen, it takes about 7 to 14 days. So that's why I say this is probably not happening first, right? It may happen later, right, or subsequent exposures to the same antigen, but it's probably not the most common way in which complement is being activated, right? Classical, as I said, because this is what we discovered first. The alternative kind of sounds like, oh, this is what happens alternatively, right? Oh no, this is probably what happens first, okay? So, classical pathway, right? The key here is antibodies bind. Complement proteins, as you can see, bind to the antibody itself, and it's going to activate this cascade of events, right? And within that cascade is the C3A being C3 being split into C3A and C3B, even C5 being split into A and B components, right? So that you get that reaction, right? That cascade, that domino effect where we get the inflammation. We get maybe C3b binding. We may get the MAC complex and cell lysis happening. So they go through the specific steps, which we're not going to worry about in this course. This is not an upper level immunology course, right? So just the beginning and the end for us, right? And a little bit of the middle, you could say. Okay, so the alternative pathway is C3, as I said, is always being split into A and B components. And then when the convertase happens, right, there's a signaling mechanism that continues this path, right? So once, like the C3bs, if they start binding, you're going to get more C3A and C3B produced, right? And then as we said, C3A, right, inflammation. C3B is that binding, opsonization helps the phagocytic cells, right? And the very last outcome that could happen is the formation of the MAC complex. So, series of proteins found in our blood and tissue fluids. They circulate mostly inactivated, right? They have to be activated, whether it be by the alternative pathway of C3 being split all the time, right? And then there's a step in that process where if those molecules are being utilized, then we're going to go forward. Or if antibodies activate, and we know that is the classical pathway. Or the last one they don't show in that animation is the lectin pathway. And what the lectin pathway is almost like a pattern recognition. There are components in the complement system that can recognize and detect mannan, which is a polymer of the sugar mannose, which is common with microbial organisms. This is a sugar complex that's common to them, not to us, right? So when the system detects that, it's going to start the cascade of events, right? So again, we're going to get that major point where C3 is being split into the two pieces, and I'll go to the next slide that actually shows it a little bit better, right? So it doesn't matter which way we start it, right? Alternative pathway with C3b binding, right, from the C3 that's always being split, or the mannose binding, lectins binding to mannose when it's present, or antibodies binding to complement proteins for antibodies that are bound to cells. It's all going to get the formation of an enzyme that's going to convert C3 into more C3a and C3b. Once that happens, notice it's a chain reaction. So A is going to elicit, C3a is going to elicit inflammation, right? The next set of conversions, major conversions, is C5 to C5a, which again is going to specifically call the neutrophils to that area. C3b, if it binds, right, it's going to help with opsonization, tagging for destruction by phagocytic cells. And then if there's a membrane, right, as you can see, the one that starts that is C5b, and then the rest of the proteins fall in line and can form that MAC complex. That is a simplistic view. There's lots of steps, right, that we're not going
to know C3A helps with getting
inflammation.
Yeah, getting that.
You got to link C3A and C5A with inflammation. C3b is our opsonizer, binding, tagging for destruction by phagocytic cells.
And C5B is the MAC attack.
Well, no, the MAC complex,
Yeah.
right, is the MAC complex, is the membrane attack complex. That's important. It only inserts into membranes, right, not into cell walls. This picture is an excellent summary of the level of detail you need to know for my course. This course. Okay, remember when we talked about phagocytosis, right? And how bacteria evade phagocytosis? Well, how is it that C3b doesn't bind to our cells, right? And we don't destroy our own cells? Well, because we have regulatory proteins on our cells that stop C3b from binding and activating complement, okay? So notice the regulatory protein on our cells, right? C3b, even when it binds, it's blocked by those regulatory proteins, so the other complement proteins cannot associate with it and cannot activate complement. A microbial host cell, right, the regulatory proteins do not bind to it normally, right? So therefore, the other complement proteins can interact with C3b and activate complement. And then, of course, this would be the alternative pathway, right, by C3b binding. But guess what? Some microbes out there have figured out how to coat themselves in our regulatory proteins and stop complement activation. Yeah.
Well, it's like when the case study with shigellosis, it gets in the cell and then it can like disguise itself.
Yeah.
So the body doesn't kill it. That's a nasty little bug.
It is. Yeah, yeah. Most pathogens, right, the reason why they make us sick is because they're tricky little, nasty little bugs, right? They've figured out how to get past our defenses, or even in this case, use our defenses against us, right? And we refer to, in general, those abilities they have as virulence factors, right? So what is it that they have, right? What ability do they have that causes them to either evade our immune system or disrupt our immune system or get past our immune system that allows them to grow, and some of them even damage us and potentially kill us, right? What makes them pathogenic? What makes them disease-causing, right? And it's these attributes, these virulence factors, these abilities, you could say, that they have.
Not to sidetrack, but the complement regulatory protein, people who have like an autoimmune disorder or rheumatoid arthritis, things like this, where there's constant inflammation going on, their body's literally attacking itself, are they missing that protein?
No, that's a different mechanism. So she asked about arthritis, how the body as an autoimmune response is attacking one's own self. She was asking if you're missing this particular regulatory protein. That inflammation process is not triggered by complement. It's triggered by other things. All right, so the next one, this is one of my favorite ones, and this is the antiviral activity of the cytokine class of interferons. And so again, this is a chemical message. Interferon's a chemical message. And so what happens is sometimes when the virus gets in, remember we have detection mechanisms inside of our cells. So they're able to detect that there's viral nucleic acid inside, and they're going to produce this cytokine referred to as interferon. And there's a couple of different interferons. It'll leave this virus-infected cell and attach to receptors on neighboring cells. And what this does is like a warning signal to the neighboring cells. It says, I've been infected by a virus, get ready, it's probably coming your way next. Because this guy can't save himself. It's too late. He's basically screaming, I'm dying. Okay? The good news, he warned his friends in the process, right? He said, the bad guys are coming, right? And that is by the interferon. So what does this guy do, right? It's a chemical signal, right? Binds to a receptor, the cell's going to do something about it, right? If somebody tells you there's a bad guy, you're going to run, hide, whatever, right? These guys can't run and hide, so guess what? They get prepared to fight, right? They produce digestive enzymes that will destroy the virus when it gets in. The sad thing is it will destroy them too, right? It will destroy them too. So it degrades the messenger RNA of the virus as well as their own. So they will die. But the good news is, what have we stopped? The virus, right? So it can't replicate in that cell, right? So we can therefore stop the spread of the virus, right? By calling out, right? And saying, hey, get ready. You're going to have to fight, right? To save the whole organism, you're going to have to fight. And that's the good news, is that we are multi-celled organisms. We can afford to lose cells, right? It won't kill us. All right, and I gotta do fever in like 2 seconds, right? So, fever, what is that? We get hot, right? We turn up our internal thermostat. Why would you want to do that?
It takes you out of the optimum temperature.
Right. We talked about how organisms, even like ourselves, have this optimum range of growth, right? But if we get too hot, what happens? We're going to denature proteins, we're going to melt membranes, right? We're going to kill that cell by heating it, right? That's why, one of the main reasons why we cook our food, although most of us like that texture as opposed to the raw texture. But, it's helpful. Also, when we elevate temperature, chemical processes happen faster, right? So we're going to respond faster as well. So our body has receptors, fever receptors, endogenous ones, pyrogens, that are going to turn up that internal temperature, okay? The bad news, though, is that some pathogens themselves produce substances that are detected by our system as pyrogens, except these are exogenous. But they do the same thing. They turn up our thermostat, okay? Bacteria are notorious for this, right? So whenever you have fever that's not intermittent, right, it's usually not your body that's turning it on, it's the microorganism. And bacteria, as I said, are notorious for this. So usually when you have a high fever that's continuing to go up, it's not subsiding, right, and it just keeps going, it's probably because you're a bacterial infection, okay? When it goes up and down, it's usually your body regulating it, or it could be a viral infection. Again, it's usually your body that's regulating it, not the organism itself, right? And that's where, you're like, oh, well, everyone's like, oh, fever's bad. Fever's bad. Because, yeah, it's bad when it's the microorganism that's causing you to go hot, where you could go hotter than you're supposed to, right? And obviously, those particular organisms, the heat doesn't affect them. Because evolutionary-wise, why would they produce heat chemicals, right, cryogens, to pump up the heat in their host and kill themselves, right? That would be pretty stupid, right? Evolutionary-wise, you wouldn't exist anymore, right, if you were always killing yourself. Make sense? So these organisms, unfortunately, right, that doesn't have an effect on them. The unfortunate thing is that fever too high for us would what? Kill us, because of the same things happening, right? If we get too high, we're going to denature our proteins, we're going to melt our membranes, and we're going to be toast, right? So fever, right, when we do it, is a normal immunological response, right? The bad news, again, is that some microorganisms can elicit fever as well, right? Where they could cause our system to go hot.
But usually when it's like the bacteria doing it, we spike high real fast.
That's what I'm saying. You spike, when it's bacterial, you spike high fast, and, it's not usually, something that gradually kind of goes along and then dips down and comes back up,
You get the hot flashes and you're not menopausal, and it's...
Yeah, exactly. We have that lovely hormonal interaction that women experience, thanks, right, that also can turn up the heat, which is not cool. So you guys understand about fever, right? It is an immunological response, right? The unfortunate thing is that some microorganisms, especially some bacteria, can turn it up too. They can use our defenses against us, again, evil bugs. So this is the it for innate. But again, a lot of these things that we've talked about are going to link into adaptive, because these aren't separate. Our immune system isn't separated, right? We just separate it for understanding purposes. I want to rework that lecture, right? I'm not happy with the flow of my adaptive PowerPoint lecture, which is why it isn't posted yet. I'm going to try and work on it today and tomorrow, and get it up by tomorrow, so that those of you guys that like to have it ahead of time, right? But I will post the objectives, because I'm not tweaking that. But the actual order of the lecture and the pictures and stuff like that, I'm not happy with the flow of it, and I want to change it. So I'm going to do it this time. I will get it done, I promise. And where's my thing? There it is.